Sure.
Okay, thank you very much, Harry. Delighted to be here. Welcome, everyone. Good afternoon. My name's Kai Yang. You can call me Kai. I'm a third-year medic at Merton College at Oxford, and today I will be covering a revision class on medical genetics. And I think, yeah, I'll go over this in a second, but the way the lecture will be broken down is we'll cover the following points in the spec. So we'll go through an overview of general concepts of medical genetics, and then we'll delve into individual kind of specific aspects. So we'll go into chromosomes, genetics of disease, genes and populations, identifying genes for specific gene defects and genetic changes associated with multifactorial diseases, and mutations in human disease, and finally, cancer genetics. Actually, can you guys see my mouse at all? I don't know, because I can see it on my screen, but I don't know if it's coming up on your screen.
Just give it a wiggle. I'm not sure I can, unfortunately.
Okay, I'll see if I can get. Oh, okay, maybe not. Okay, we'll have to get by. But if anything is unclear, then just do let me know. And yeah, so with these concepts that we'll be covering today, the way I'm going to try and break it down is we'll spend 2 or 3 slides looking at a theory and particularly kind of high yield areas within each topic. We'll go for 1 slide for practice part A style questions. So that's kind of our short, snappy, multiple choice questions. And we'll finish off for each topic, or for most topics, with 1 slide. We'll take a part B question. So that will be kind of an essay type question. And then we'll dissect that and talk about how you might structure those sort of questions. Okay. And at any point, if you want to raise your hand or kind of leave a question or even an answer in the chat function, then please do. Okay, so just to go over some general concepts of medical genetics. So hopefully you'll be familiar with some of these, but no worries if not. So a lot of diseases, most diseases in fact, will result from the interaction between the genes and environment. Nevertheless, you do get some phenotypes and some diseases which are either primarily determined by genetics, such as achondroplasia, or the environment, such as lead poisoning. And these tend to have an actual lower overall burden of disease at the population level. And this is compared to phenotypes which require or are associated with both a genetic and environmental elements. So the classic example is many chronic diseases, such as type 2 diabetes, and these kind of conditions which factor in both of these aspects tend to have a higher burden of disease at the population level. So just to go over some basic definitions, which will be kind of the foundation of a lot of what we cover today. The word phenotype is can describe the physical manifestation or description of a character in an individual organism. So if we go back to Gregor Mendel and his work on peas in, I think, the 19th century, the characterization of a pea is green. That is the phenotype of that pea in that respect. And then on the other hand, the genotypes are the genes that an individual has or an organism has at a particular locus, and they are subsequently responsible for the phenotype observed. And linking nicely to that is a gene, which is just a section of DNA nucleotides which encodes a polypeptide. And so they specify the phenotype at both a gross morphological, kind of a gross level in terms of the morphological characteristics, but also at a molecular level in terms of the protein products, the RNA products, etc. Alleles are versions of genes, and we'll be talking a lot about alleles later on and how we can perhaps measure different levels of alleles and consider the different dominant and recessive forms of alleles. And finally, penetrance is the chance that a given genotype will cause a particular phenotype. So namely, it's the chance that if you have a mutation that you'll get a disease. So that's a general overview of medical genetics and the key terms. And now we'll delve into individual details of the topic. Okay, so first of all, chromosomes. So with chromosomes, hopefully you can see these diagrams. I'm sorry my pointer doesn't appear to be working, but we should be able to kind of work through it together. So with the chromosome itself, if we go from top to bottom in the diagram, you can see it consists of 2 identical sister chromatids, and these are joined at the centromere region, as we can see here, which is described as a constricted point of the chromosome, which joins the 2 sister chromatids. The chromosome itself is characterized by a P arm, which is the short arm of the chromosome. So you can remember that as P for petite, meaning small, and then the Q arm, which is the long arm of the chromosome. And these are comprised of DNA molecules. Some other points to note, which often do come up in exams, especially Part A questions, are the telomeres, which are the ends of the chromosomes. And I'm sure you'll be aware of the role of telomerase in some cells to kind of preserve these telomeres and prevent the shortening of chromosomes. And another aspect that's important is if we look at this bottom left image, we can see that chromosomes can be classified according to the location of the centromere structure, which we talked about just now. So classically in humans, we have acrocentric, submetacentric, and metacentric chromosomes, and these are all governed by where the centromere is located. So to put it in brief, acrocentric chromosomes are where the centromere is towards the end, submetacentrics is when the centromere is just off middle, and metacentric is when the centromere is kind of in the middle. And so this, once again, is a key concept that will come up time and time again in your Part A questions. So in terms of chromosomal abnormalities, there are two main types, two main brackets that we can consider. So we've got structural and numerical abnormalities. And so firstly, if we consider structural, these tend to involve a rearrangement of the chromosome and often entail breakages and reconstitution of the chromosomes in an abnormal fashion. So these structural abnormalities, if we start with translocations, these can be both balanced or unbalanced, and this refers to whether the amount of genetic material, the total amount of genetic material, is normal or whether it's changed. If it's normal, if it remains normal after a translocation, it's balanced, and if it doesn't, it's unbalanced. Now, what do we mean by translocation? Well, a translocation event is when genetic material is transferred from one chromosome to another and this can happen, I guess, at different stages during mitosis or a cell cycle. And in terms of the two main types, we have, like we said, we have a balanced translocation and an unbalanced translocation. So if we go through some examples, a Philadelphia chromosome is an example of a balanced translocation, and it's common in chronic myeloid leukemia patients. And what happens here is if we look at the diagram on the bottom right, we can see that we have a breakage in a long arm from chromosome 9, and it gets transferred to it becomes controlled by the breakpoint cluster region of chromosome 22. And the reason this is kind of clinically significant is that the ABL, which encodes an oncogene, becomes kind of uncontrollably expressed by this breakpoint cluster region promoter area, and this forms a kind of chimeric protein which is oncogenic and ultimately drives the chronic myeloid leukemia. So that's an example of a balanced translocation, and it's important to note that even though it's balanced, it can still have harmful effects, like we've discussed. Another type of translocation is a unbalanced translocation. An example of this is a Robertsonian translocation. So this occurs when we have translocations whereby the breakpoints are at or close to the centromeres of acrocentric chromosomes. So remember, we have acrocentric chromosomes here in this diagram in the bottom left. And what happens here is you get fusion of the long arms, and the short arms are lost. So you can see that because we're losing the short arms in such a case, it means that it's an unbalanced translocation as we're losing genetic material. So what are the other structural chromosomal abnormalities? Well, we have duplications, deletions, and insertions. Hopefully these are all fairly self-explanatory, and we will go into a bit more detail on some of these in our practice Part B question in a minute. So in terms of numerical abnormalities, these are categorized as kind of aneuploidy, monosomy, and trisomy. And in terms of aneuploidy, that's just referring to a state whereby there's an abnormal number of chromosomes within a cell. And so monosomy refers to the fact that, or refers to the state whereby there's one rather than two copies of a particular chromosome within a cell. It's important to note that monosomy of an autosomal chromosome, so chromosome 1 to 22, is almost always fatal, but there are quite commonly monosomy conditions of the sex chromosomes, which we will come across in a minute. And finally, trisomy is just the presence of 3 rather than 2 forms of a particular chromosome. And there are 3 main conditions which we tend to associate with trisomy, and we'll cover those as well, or we'll go over those as well. So how do we analyze chromosomes and changes in chromosome structure or chromosomal abnormalities? Well, traditionally, it's been done through this process called G-banding, whereby chromosomes are stained with a G-EMSA dye, and so this is a DNA binding dye. And the way it works is that active areas are stained light, and this is because the chromatin itself is less condensed, so it's more likely to be transcribed. And then subsequently, the chromosomes can be arranged and analysed by size on the karyogram. Another method that has developed is something called FISH, so it's fluorescence in situ hybridization. And this is when a single-stranded DNA probe anneals to a known complementary target sequence. So it's especially good if we're looking for something which we kind of suspect, or we have a known target that we want to look for. And so it can be used to identify chromosomal abnormalities such as trisomies, so numerical abnormalities, but also structural events such as translocation events. And interestingly, it's also been used clinically. So in breast cancer patients, we can detect overexpression of HER2, so HER2, Herceptin 2 receptor, and this can indicate the suitability of such patients to Herceptin therapy. And finally, another way of looking at chromosome complement is something called array CGH, so comparative genome hybridization. And this is particularly useful to look at regions of gene amplification or loss. So it's commonly used in cancer patients and also dysmorphic individuals, so individuals perhaps with an unusual phenotype. And if you can see the diagram here, in short, the way CGH or array CGH works is we label both test DNA, so that's the patient DNA here in red, and the control DNA, which is the DNA of someone of a wild type person who doesn't have a given disease. And then we allow both of these labeled DNAs to hybridize with microarray wells, each of which contains a particular DNA probe referring to a particular locus or a particular gene. And then we can wash away the unbound DNA. And then subsequently, by using computerized fluorescence imaging or analysis, we can then see whether there's predominantly green or red spots at particular locations. And if there's red dots, in this case, this indicates a gene amplification event. So there's more DNA at a particular locus in the patient where there's a red dot, and vice versa for the green dot. And if there's a yellow dot, this indicates there's no amplification or loss at a given locus or a given site in the patient DNA compared to control DNA. So yeah, these are three different ways, I guess, of looking at chromosome complement. Each of them have their own benefits, and often they can be used in combination to complement each other. Okay, so I'm now going to cover some practice questions. So this will be very good practice for your Part A multiple choice questions. Some of this we haven't actually covered in the revision session, but you might have come across already. And if not, we'll go through it either way. So we'll go through the questions individually. If you would like to give it a go, then please do feel free to unmute yourself or type an answer into the chat. And if you don't know, if you don't want to answer, that's fine. We'll give it a minute and then I'll go through the answers. So the first question is something we've covered. So what are the three main types of chromosomes we have in humans? And then how are these three main types of chromosomes differentiated? So what structure, the positioning of what structure differentiates these three chromosomes? Anyone want to give that a go? No worries if not. We can go through it together. So in terms of the three main types of chromosomes, we have the acrocentric, submetacentric, and metacentric chromosomes, and they're all defined and classified by where their centromere is located. So acrocentric, the centromere is at the end, submetacentric is just off the middle, and metacentric is in the middle. And so if we go through some examples of disorders caused by aberrations in sex chromosomes, hopefully you would have come across some of these. The main two or three, I guess, we tend to consider are Klinefelter syndrome, which is whereby there's a genotype of XXY. So males have two X chromosomes and a Y chromosome. We also have Turner syndrome, which is categorized as 45X. So it's females with just one X chromosome. And we also have triple X syndrome, or triple X, which is 47XXX. So a female has three X chromosomes. So these are all three kind of conditions which are good to know about, because they're both clinically relevant and also for the purpose of your exams, they will crop up as very easy, hopefully easy to answer, short MCQ questions, MCQs. Okay, and our final two questions on this topic. So true or false, X-linked genes in males are haploids? I'll give you a second to think about that. So the answer is true, and it simply refers back to the fact that males have one X chromosome and one Y chromosome, unlike females who have two X chromosomes. And on a similar note, true or false, females have 2 X chromosomes and so express twice the amount of X chromosome gene products. So have a think about that, and then we'll go through the answer in 10-15 seconds. Okay, so the answer for this one is false. And this is because of a process whereby in females, all but 1 X chromosomes are inactivated in cells. And these excess X chromosomes are found in Barr bodies. So normally, if you have 2 X chromosomes in a female, 1 chromosome will be found in the Barr body. But it's also important to note that if we have, for instance, a female with 4 X chromosomes and they have a 48 XXXX genotype, they would have 3 Barr bodies with 3 inactive chromosomes. So this means that the same amount of products of X-linked gene products are found in males and females, and this is a phenomenon referred to as dosage compensation. So it's important to know these because these are the short, kind of snappy facts that might turn up in your Part A's. Okay. So finally, on the topic of chromosomes, we'll just go over a past Part B question and how we might want to go about it. So this is an example of a question that's been thrown in the past. It's called give examples of how disorders that result from structural and numerical human chromosomal abnormalities arise. So if I would, I'll talk about how I want to go about it. Unless anyone has any thoughts, please do let me know. Let me know via the chat or just unmute yourselves. If not, we can talk through it. I'll talk through it with you guys. So with this question, it already breaks it down quite nicely for you into structural and numerical human chromosomal abnormalities. So we immediately can kind of categorize the events into structural and numerical. What you then do with it from there is kind of up to you and for almost personal taste, in the sense that you could just have two paragraphs or 2 or 3 paragraphs on structural abnormalities and then 2 or 3 on numerical abnormalities. You could look to compare and contrast them, etc. And so the way I would perhaps recommend go about it, it's not the right way, but it's one way of going about it, is breaking down these kind of abnormalities and the chromosomal events that underpin them. So firstly, I would consider what processes cause specific chromosomal abnormalities. So I'll talk about trisomy in a minute and how trisomy arises due to non-disjunction, what disorders these then lead to, and finally, how the chromosomal abnormalities cause a disease and the associated symptoms. Because if we look at the question, give examples for how disorders that result from structural and numerical human chromosomal abnormalities arise, you could very reasonably argue you perhaps want to give insight into how the chromosomal abnormalities arise in the first place, but also how these abnormalities then cause the disease and the symptoms. So let's go through 3 examples. So trisomy, you should hopefully be aware of the different forms of trisomy that can occur. In particular, we get trisomy of the smaller chromosomes because trisomy of large chromosomes isn't associated with cellular viability. And of course, chromosomes are organized based on size, so 1 being the biggest, 22 being the smallest out of the autosomal chromosomes. So trisomy 13, Patau syndrome, trisomy 18 is associated with Edwards syndrome, and trisomy 21 is associated with Down syndrome. And in terms of characterizing how these chromosomal abnormalities arise, they occur through non-disjunction in either meiosis 1 or meiosis 2. So in meiosis 1, you get a pair of homologous chromosomes which fail to separate, and therefore the gamete contains both homologs for 1 chromosome pair. And in meiosis 2, we have sister chromatin failing to separate, and thus gametes contain 2 copies of 1 homolog. And so that's addressing this part, what processes cause the chromosomal abnormalities. And now to address how these chromosomal abnormalities cause the disease, we can then consider the example Down syndrome, which is often associated with a kind of propensity to develop Alzheimer's. And this can be elucidated by the fact that there's overexpression of overexpression of the APP gene on chromosome 21 because we have an extra copy, and the APP gene leads to increased amyloid beta protein, and this triggers abnormal and aberrant synaptic pruning in the brain, so it leads to aberrant neuronal function and neuronal formation, and this can potentially elucidate the Alzheimer's seen in Down syndrome patients. So you can see with this example how we've gone from looking at how a chromosomal abnormality arises, the disorder associated with it, and finally how the chromosomal abnormalities then cause the disorder. And similarly, we can do this with deletion events. So a microdeletion are deletions less than 5 megabases, and an example is DiGeorge syndrome, which is characterized by haploinsufficiency of TBX1, and by that we mean that one copy of the gene is insufficient to retain the function needed for the kind of cell to, or the organism to function normally. And so haploinsufficiency, an insufficient amount of TBX1, means that the formation of many tissues and organs is aberrant and abnormal, and this kind of manifests in heart defects, an underdeveloped thymus, and palate defects. And so here are just 2 examples, and then finally we talked about reciprocal translocations with the example of the Philadelphia chromosome. So hopefully we can I've been able to show that quite a daunting question potentially at first can be broken down in a logical fashion, and we can actually approach it in a kind of systematic way that can also incorporate some perhaps extra points that aren't covered within the main syllabus. Okay, so are there any questions, by the way? If so, do let me know, and feel free to use the chat function. And so if not, we'll move on to the next topic, which is the genetics of disease, and we'll also combine it with 3.7 in the spec, which is mutations in human disease. Okay, so just an overview of the genetics of disease. When we're talking about single gene disorders, which I guess there's a variety of diseases caused by these single gene disorders, this is referring to mutation in a gene that encodes structural protein, which subsequently leads to either no production or abnormal production of a non-functional protein, or of a protein, sorry. So yeah, no production or either production of a non-functional protein. And so a lot of these single gene disorders are associated with either single base changes, otherwise known as point mutations. And then we'll use this graphic, this kind of cartoon or image on the left to help talk us through. So in terms of point mutations, point mutation events, these are typically categorized as substitution, or they can also be indel events. So in terms of substitution mutations, these can be transition or transversion mutations, whereby in a transition we go from a purine to a purine, so a G to an A or vice versa, or a pyrimidine to a pyrimidine. And in transversion mutations, we go from purine to pyrimidine or vice versa. These substitution events can either be synonymous in the sense that they don't change the amino acid sequence, and this is because of redundancy. So what we mean by this is some amino acids are coded for, some residues are coded for by more than one codon, and therefore changing one amino acid might not actually change the amino acid sequence. And therefore this leads to what we call a silent mutation, which we can see on the diagram on the left in the second line. We've changed a nucleotide, but the amino acid remains the same compared to our normal sequence. On the other hand, non-synonymous mutations do change the amino acid sequence, and they can either be missense mutations or nonsense mutations. So if we look at the diagram on the left again, by nonsense mutation, what we mean by that is a codon specifies an amino acid which is replaced by a stop codon. So you get a premature termination codon and you get a truncated polypeptide being formed. So that's a nonsense mutation. A missense mutation is when we replace one amino acid with another. And so this is what we can see here. We can see it's just one codon that's been changed and just one amino acid that's been changed. And it's quite interesting. Sometimes this can have a very minimal effect, but if there's residue change occurs in perhaps the active site region of an enzyme, it can have quite catastrophic effects. And finally, with frameshift mutations, with indel events, so this is when we either insert or delete a base. It can be a base, it can be more than a base, but in the context of point mutations, if we insert or delete a single base, this completely changes the read-in frame. So what we mean by that is if we look at this diagram on the left, we can see that once we've inserted or deleted a base, it completely changes the amino acid code that is produced. And this is because if you can imagine, if we insert one nucleotide, the whole sequence gets shifted along because we still read in the codons in groups of 3, but we're reading a different frame each time. And so these tend to be fairly catastrophic and actually often lead to nonsense mutations. And so it's really good to be familiar with these key definitions and these key terms that we've covered on this slide, We'll get a bit more practice in a minute, but these are very much your classic Part A sort of questions. Okay, so it's a bit more serious. I'm sorry it's a bit heavy, but this is once again really good, useful information to know. And some of it is just learning some facts, but a lot of it is if you understand especially when it comes to inheritance, if you understand the principles, then you won't actually need to learn the facts off by heart. So we'll go through these right now. So in terms of modes of inheritance, we'll cover 4 modes. So autosomal dominant, and examples of these are Marfan syndrome, achondroplasia, Huntington's disease, and these do affect the heterozygotes. And the reason is that, and by heterozygotes we mean that individuals who carry one kind of mutated allele, so to speak, or and one normal allele, and this is because only one abnormal or mutated gene is required for the disease to manifest, and the chance of passing this mutation on to offspring is 50%. Secondly, with autosomal recessive conditions, so these include cystic fibrosis, sickle cell anemia, the chance of, so this is when 2 copies of an abnormal gene are required for the disease to occur. This is often associated with consanguinity, and what this means is that this is breeding between people who share an ancestor, so people who are, namely people who are kind of related. And the reason this increases the risk, or this is associated with autosomal recessive genetic conditions, is because related partners are more likely to have mutations in the same gene. And in terms of the chances of 2 carriers having an affected child, that's 25%. And if you draw your kind of the genetic plots, I think they're called, you can kind of map that out, but hopefully you guys will be familiar with that. So if we now turn to X-linked disorders, so once again we can have X-linked recessive or X-linked dominant disorders. So examples of X-linked recessive conditions are haemophilia, colorblindness, and Duchenne's muscular dystrophy. In such cases, the females will be carriers, but they will be unaffected because of the X chromosome inactivation that we talked about earlier. But it's important to note that sometimes this X chromosome inactivation may be skewed, in which case females might be affected. And so typically, if a mother passes on the mutated X chromosome to a daughter, that girl would also have a backup X chromosome from the father, and so she'll be a carrier. So the risk to the female offspring is 50% carrier, 50% normal. Alternatively, for males, because they're hemiazygous, the genes in the X chromosome, i.e. they've only got one X chromosome and no backup X chromosome, the risk to male offspring will be 50% affected, 50% normal. And finally, X-linked dominant conditions, an example is Rett syndrome. Females are affected in the presence of one mutant X chromosome, although they tend to survive due to the X chromosome inactivation that we talked about just now. And so the risk to the female offspring is 50% affected, 50% normal, and the risk to the male offspring is 50% very severely affected because they don't undergo this X chromosome inactivation, and 50% normal. So there's a lot of content on that slide, I know, but hopefully if you perhaps give some time to think about it, it will all make sense. But a lot of these, especially the examples of different conditions, so for instance, knowing that Rett syndrome is an X-linked dominant, these are frequent questions that will come up on multiple choice questions for your Part A exams, so they're very good to know. Okay, so we'll hear some practice questions. Once again, I'll open it up for you guys. If you want to answer, please do, but if not, we'll go through it together. So the first question, or first, I guess, fill in the blank, is a mutation in the AAG codon is changed to the ACG. It's changed to the ACG codon. This is an example of what sort of mutation. So if you want to give it a go, please do, but no worries if not. Okay, so this is an example of a missense mutation because we've changed one codon. We haven't necessarily changed the reading frame, nor have we encoded a premature stop codon, so it's a missense mutation. Secondly, Fragile X syndrome is an example of a triple repeat expansion. So we haven't covered it just now, but we'll cover it now. So trinucleotide repeat sequences are quite common within the genome, and they can become unstable during replication, which leads to the amplification and generation of more repeats in a process called expansion. And this subsequently often if you surpass a particular threshold for repeats, this can disrupt protein function. These expansion events tend to kind of occur progressively down generations, and this leads to more severe phenotype risks of successive generations in a process called anticipation. So that's an example of a trinucleotide repeat expansion. Another one would be Huntington's disease, which also, once you reach a particular threshold, then that's when the disease tends to manifest prominently. Okay, and thirdly, inheritance of mutations in the mitochondrial genome is often associated with disorders such as, so the answer here are neuromuscular diseases. So these tend to involve impairment of the function of the muscles, or so the kind of the functioning of muscles. And so two examples are MRF, so myoclonic epilepsy with ragged red fibers, and also MELIS, which is mitochondrial encephalopathy, lactic acidosis, and stroke. And so it's important to note that maternal inheritance of the mitochondrial genome occurs. So it's all but kind of inherited from the mother, and so therefore these diseases are transmitted through the mother. And finally, deletion of 4 BP, so base pair, deletion of 4 base pairs would result in a frameshift mutation, because only if it's a deletion or insertion of a multiple of 3 will it not change the nature of the frameshift, and that's simply because the codon sequence is read in groups of 3. The code is read in a sequence of 3 bases. So if it's any number that's not a multiple of 3, it's going to change the way the sequence is read. Okay, so we've got, I think, 2 more topics to cover. So we're halfway through, but we'll try and keep it, or we'll try and get done for 5. Okay, so we're now going to cover genes in the population. And so we're going to cover the Hardy-Weinberg equation. So the Hardy-Weinberg equation is a mathematical equation that can be used to calculate genetic variation of a population at equilibrium. And here it is here. We'll talk about it a bit more in detail. Here it is in the bottom right, sorry. We'll talk about it in a minute. And so it's based on this principle that for diploid sexually reproducing organisms, the allele and genotype frequencies remain constant from generation to generation under given conditions. So I guess you could say these are the assumptions, or this is the kind of the premise of the Hardy-Weinberg equation. So firstly, we're dealing with a large population, and therefore this kind of negates genetic drift. Secondly, there's random mating occurring between individuals. For thirdly, there's no migration, so this prevents gene flow into or out of the population. There are no net mutations, no selection pressures for or against a genotype, and there are only 2 alleles for a given gene that's meant to be, within the population. So here is the equation. So P and Q, so P is referring to the frequency of the dominant allele in the population, and Q for the recessive allele, and subsequently a P squared notation is referring to the chance of being homozygous for the dominant allele, Q squared is homozygous for the recessive allele, and 2pq is the chance of being heterozygous, so a carrier. Okay, so it's important to note that although the Hardy-Weinberg equation puts out quite nicely a prediction of how assessment of the gene, the proportion or frequency of genes within the population, these conditions don't actually occur in real life in human populations, and this is for several reasons. So firstly, there tends to be assortative or non-random mating. So an example of this is there tends to be a correlation between partners finding a partner of similar intelligence, and so this is an example of non-random mating. And similarly, for rare recessive traits, heterozygotes or heterozygote individuals are more likely to mate than expected by chance random mating. So you can see that this does not always occur. The idea of random mating does not always occur as is specified by the Hardy-Weinberg equation. Secondly, selection. So I'm sure you'll be familiar that certain mutations can either increase or reduce the reproductive fitness of a carrier, and only if it increases then the fitness, then positive selection will occur, and vice versa. Thirdly, mutations are a very common source of variation, and these can be both detrimental and advantageous, and this links to this idea of selection, which we discussed just now. So next up, we have genetic drift as another factor for why ideal conditions assumed by Hardy-Weinberg equation aren't necessarily met. And by genetic drift, this is just referring to the process whereby allele frequencies can change, and this is prominent in smaller populations due to the increased reproductive fitness of some organisms, perhaps due to selection pressures, and therefore the allele frequency in these organisms is unlikely to be the same as in the whole population. And an example of genetic drift is the founder effect, which occurs in small, recently isolated populations, and these tend to have reduced genetic variation compared to the original population. And finally, migration, which leads to gene flow and movement of alleles from one population to another, and this will be particularly prominent and have great effects if the population is small and if the allele frequency differences are large. Okay, so let's just move on to some questions. So this one, I can give you a bit longer. We'll go through it together, but this is an example of a question you might get. So if we say the allele Y, so the recessive allele occurs with a frequency of 0.8 in a population of humans, what is the frequency of the genotype YY, so the heterozygote phenotype? So you try using the equation we had just now, this one, to work it out. I'll give you the answer in a minute, but I'll give you a minute if you want to have a go at it. Okay, so we'll just talk through it briefly. So if we label, sorry, apologies, I would have perhaps used a whiteboard function if this can allow me to, but I don't think I'm able to do that right now. But if we label the recessive allele Y, if we insert that into our equation here as Q, and then the big capital Y as P, so the dominant allele, then we can form the equation Q, which is 0.8 plus P equals 1. So P equals 0.2, Q equals 0.8, and then if we refer back to the equation, this heterozygote genotype 2PQ is simply 2 times 0.2 times 0.8, so our answer here would be 0.32 as the frequency of the genotype capital Y, small y. And secondly, in terms of why there might be ethnic differences in disease frequencies, this can arise through multiple reasons. So we might depend if it's an open or closed population. There might be certain survival advantages, perhaps based on different selection pressures imposed or the different variations within populations, and also consanguinity, which we discussed just now. So an example of that would be the blue fugates, which were a population in, I think, in North America somewhere. And what happened is that there was lots of consanguinity within this population, and these people were characterized by a genetic trait that led to something called methemoglobinemia, and this was excessive methemoglobin within the blood, and this doesn't bind oxygen very efficiently, and therefore this led to these individuals having kind of almost like a blue-like skin, and this was kind of particularly prevalent within this population because there was lots of consanguinity. Okay, so I think I was mistaken. Now we've got 2 more chapters or sub-chapters, so to speak, to cover, but we'll try and get it done in time. Okay, so our penultimate area to cover is 3.6 in the spec, referring to identifying genes for single gene defects and genetic changes associated with multifactorial diseases. So the underlying foundation for this topic is this idea of DNA polymorphisms. So polymorphism markers are genetic variants that occur at high frequencies within the population, although they themselves are not pathological. So here are 3 examples. So SMP, so single nucleotide polymorphisms, occur due to substitution, insertion, or deletion of a single base in non-coding regions. Subsequently, we also have variable numbered tandem repeats, and this is changes in the numbers of repeated DNA sequences arranged in tandem arrays. and these tend to be repeats between, I think, 10 to 100 nucleotides in length, and they are all clustered and kind of orientated in the same direction, hence kind of tandem arrays. And thirdly, we have microsatellites, which are a class of VNTRs that have repeat units 1 to 6 base pairs in length. So they're a bit smaller, so we can differentiate these based on differentiate the VNTRs. So the VNTRs also refers to as mini satellites and microsatellites based on the length of these repeat sequences. And so sites of polymorphism can be used to indicate whether a particular allele, identified by its polymorphism, segregates with the genetic condition. And so the location of the marker that segregates with the condition also assists in pinpointing the site of the mutation that causes the condition. And so this lays the foundation for genetic linkage analysis. And so typically alleles at loci on the same chromosome, they may assort independently if there is crossover events between them in meiosis. And therefore, and so if there is no crossing over between genes or between loci, then these genes are inherited together and therefore they're classified or characterized as being linked. In cases where loci assort independently, then this means that the inheritance of one allele will have no effect on the other. However, going back to this idea of linkage, the closer two loci are on the same chromosome, the greater the possibility that they will be linked. And this is a really important concept for linkage analysis and linkage studies, which we will talk about now. So for single gene disorders, to kind of unravel them and to elucidate the genetic basis of them, we use principles of linkage and linkage analysis and polymorphic markers to identify sites of mutations responsible for a given disease. And so to perform this, we genotype family members for a range of polymorphic markers across the genome, and then whether or not the markers segregate with the condition can indicate whether a disease-causing gene is linked to that particular marker region. And this then defines the maximal region of linkage. And subsequently, we can use different databases to identify a list of genes within this region of the genome, and that's how we can identify what we call kind of candidate genes for the mutation, which are kind of genes which we suspect may be linked to the disease. For polygenic disorders, the inheritance patterns are a bit less clear, a bit more obscure, primarily because it's not just one gene involved. And so the way we go about it is traditionally the way that this has been tackled is we look for siblings that are concordant for the disease, and they're analysed to determine whether there are loci which they share more frequently than the 50% expected by chance. So elevated sharing of a given region suggests that the disease locus may be close to a given marker in siblings who both have a polygenic disorder. And subsequently, the maximum region of linkage can be identified, like we've talked about before, and a causal polymorphism can be found. Nowadays, we tend to use GWAS. So you might have heard of it before, but so genome-wide association studies, and what these allow us to do is they allow us to scan the entire genome using a high density of polymorphic markers, and subsequently they allow us to carry out analysis. So we'll do a couple of practice questions. So we covered this just now, but what is GWAS? Well, GWAS is a way of looking at kind of genetic variants across and comparing them across the entire genome. So we can then identify common alleles that might be contributing to polygenic disorders in population studies. And the main advantage of GWAS is that we don't necessarily need a prior hypothesis going into the study. So you often have, you can identify common alleles contributing to genetic diseases within population studies, and you can subsequently stratify kind of populations from the study. So you can look at particular diseases, particular variants, perhaps particular ancestries, and so forth. And subsequently, another term you might have heard is whole genome or whole exome sequencing, and we often use this in linkage analysis to look at, to perhaps identify or differentiate between candidate genes and actually identify a given candidate gene. And compared to GWAS, whole genome or whole exome sequencing, which is kind of just as it sounds, sequencing the whole genome or all the exomes, They can be used especially in rare diseases where there might not be sufficient patients to consider for GWAS studies, because these GWAS studies require a lot of patients and a lot of people to provide them with sufficient statistical power. And so these whole exome genome approaches can be used to look at rarer genetic variants. And they can be applied clinically as well to try and find an appropriate drug target, especially in kind of personalized approaches to medicine. Okay, so we'll just go over a practice part B question that has come up before and how we might go about it. There's some water, sorry. Okay, so the question goes as follows. What types of sequence variations are found in the human genome, and how can these be identified? To what practical uses can they be put in medicine and forensics? So one way to go about it is to break down the DNA polymorphisms, the different ones that occur. So if we do that, first of all, if VNTRs, which we've mentioned before, they actually are the basis of genetic fingerprinting experiments, and are used in criminal investigations to identify, I guess, to match a potential person to a crime scene, and also paternity tests. So the principle of these is we can genotype people using PCR followed by gel electrophoresis to identify variations in the length of VNTRs between individuals. And so this is exactly what we can see here on the right in the diagram. So M for mother, C for child, F1, F2, F3 representing 3 potential fathers, and then we need to try and work out who is the correct father. And so in inheritance matching, we want to make sure that all the VNTR alleles must follow the patterns of the inheritance. So what we mean by that is for identifying the kind of paternal adult, so the father, the child must have alleles that match either parent. So we can see here that the child has, the upper band of the child matches a band of the mother, and the lower band of the C, the child, matches F2, meaning F2 is the father in this case. Microsatellites, which we talked about before, they're clinically relevant. So for instance, in Lynch syndrome, mutations in DNA mismatch repair genes such as MSH2, MNH1 lead to microsatellite instability. And we can identify this by looking for expanded microsatellites in tumor cells. And this can be clinically relevant because it can predict the outcome of certain cancers. So for instance, there have been reports that higher levels of microsatellite instability are associated with a better prognosis, so a better outcome for colon cancer. And then we've got SNPs, which we talked about in the context of GWAS, and also something called restriction fragment length polymorphisms. So they're actually, I think they're not covered in the core part of the spec, but these essentially are mutations which occur in restriction enzyme cutting sites, and therefore this produces different lengths of fragments which can be identified using Southern blotting. And then if we consider 2 examples of where these sequence variations may come into play, we have monogenic disorders. So for instance, the NHS 100,000 Genome Project was a project which kind of sequenced 100,000 genomes to kind of enhance our understanding of genetic variants that ultimately can help us understand new treatments, but also help with diagnosis. And this is based on, and this has been especially useful, for instance, in the context of Gorlin syndrome, which predisposes to skin cancers, and it's been helpful to perhaps identify the genes and the mutations involved within such conditions. And similarly, with polygenic disorders, we can characterize them and learn about them through the sibling pair method, which we've talked about, but also through GWAS studies. And there will be an absolute, probably over 1 million GWAS studies that are out there. So if you want to include or reference one, then please do in your essay. And one example that I've come across is this study by Uda, 2008 et al. And they did a GWAS study on patients in Sardinia. And they wanted to look at how certain polymorphisms were associated with better prognosis in patients with either beta thalassemia or sickle cell anemia, so mutations in the HBB gene. And they actually noticed that there's a polymorphism at a gene outside HBB, but actually at BCL11A, which is associated with controlling levels of fetal hemoglobin. And this is important and kind of functionally relevant because fetal hemoglobin levels are associated with better prognosis in these patients. So GWAS studies can be used to perhaps help us indicate which sequences or which variants may be associated with a given phenotype, but often you need subsequent functional analysis to validate this. Okay, we're doing well. We've got 7 minutes and we've got one more topic to cover and then we're done. So, cancer genetics. So just an introduction, I think you'll all be hopefully all aware of the terms oncogenes and tumor suppressor genes. So oncogenes can be regarded as an on switch. They promote cell growth and the cell divide and signaling cascades. So examples are growth factors, DNA binding, nuclear factors, and cell cycle factors. In such cases, one gain of function mutation can make an oncogene, can turn a proto-oncogene into an oncogene, whereby it's then unresponsive to its typically kind of tight regulation, and this can often manifest in accelerated cell division. These tumorigenic events, which whereby oncogenes are implicated in, include amplification events. As you can imagine, we get overexpression of an oncogene, point mutations, and also translocation events. And on the other hand, we also have tumor suppressor genes, which can be regarded as an off switch. So they prevent cancers by arresting the cell cycle, they induce apoptosis, and they ensure accurate replication and repair of DNA. So examples would be the BRCA1 and BRCA2 genes, which suppress tumors, which are DNA repair genes, which don't actually halt the cell cycle, but ensure that the DNA is kind of intact and not broken or damaged. And with tumor suppressor genes, both copies of tumor suppressor genes need to be mutated for the cancer to manifest, i.e. loss of function mutations. And these tumorigenic events include deletions, point mutations, but also methylation events, which actually can silence. So it's an example of an epigenetic modification, which silences the expression of a tumor suppressor gene without actually changing the sequence of the gene. So a fundamental principle in kind of cancer biology is Knudson's two-hit hypothesis. So this was discovered by Knudson by looking at retinoblastoma patients, and they noted that the age of the onset of bilateral cases was tended to be earlier than there tends to be differential onset of bilateral cases compared to unilateral cases, and this led Knudson to suggest that bilateral cases may already have one mutation within the gene responsible for retinoblastoma, and therefore there's double hit events whereby you require 2 mutations of a tumor suppressor gene for the disease to manifest, and in bilateral cases of retinoblastoma, 1 copy was already knocked out. So bilateral cases tend to be heritable, unlike unilateral cases. If it sounds a bit confusing, hopefully we'll go through it now and that will clarify it. So a cell initiates a tumor when it contains 2 mutant alleles for a tumor suppressor gene. So individuals with germline tumors in a tumor suppressor gene are predisposed to developing cancer at a younger age compared to the general population. So if you can imagine, if we need 2 hits for a given locus, if one's already deleted through a germ or mutated through a germline mutation, then there's a greater chance that a cancer is going to develop sporadically. There's inherited cancer susceptibility compared to requiring a sporadic cancer whereby you need kind of two hits or 2 mutations. Nevertheless, some tumors don't follow this rule. So for instance, methylation might inactivate the other gene, not necessarily a germline mutation. And also haploinsufficiency, which we covered earlier, may actually be enough to provide a growth advantage in some cases. So sorry if that sounded a bit confusing, but hopefully that will make sense if you go back over the slides, but if not, feel free to email me about it. Okay, so we're nearly there. This is our second to last slide. So we'll just go through some questions. So what factors may indicate a familial predisposition to bowel cancer due to Lynch syndrome? So I know we haven't covered this today in the content, but hopefully you might have come across it in the lectures this year or some of the revision. Well, the answer to this is there's several factors. So number 1, if lots of people in the family are developing bowel cancer at the age of less than 60, or if people in the family have had developed bowel cancer twice, so i.e. after the first tumour's been removed, or thirdly, if there are presence of other cancers in the family history, such as the ovary, the stomach, endometrial cancer. This may indicate a familial predisposition to bowel cancer due to Lynch syndrome. And subsequently, how can we differentiate Lynch syndrome, familial adenomatous polyposis through colonoscopy? Well, these are both autosomal dominant diseases, but the way we can characterize or differentiate them is to look for polyps using colonoscopy. So they're not associated with Lynch syndrome typically, although in some cases they might be seen. They're not seen as frequently as they are in familial adenomatous polyposis. So the presence of polyps is the key here. And finally, true or false, bowel cancer tends to develop from polyps initially, although most polyps do not develop into invasive cancers. That is true. So the transition from small adenomatous polyps to invasive cancers often takes kind of 5 to 10 years, and it typically involves the accumulation of several mutations in oncogenes and tumor suppressor genes, such as RAS and P53. So it's not just a single event that necessarily leads to these invasive cancers forming. Okay, and finally, so this is an example of a practice or a Part B question, and it reads like this. So what factors in the family history would lead you to suspect transmission of a mutation in BRCA1 or BRCA2? Discuss the implications and options available to a 30-year-old woman who's now found to carry a BRCA2 mutation. So in terms of the factors we'd consider, we'd want to consider if there are perhaps many family members with it, if there are family members diagnosed with who've been diagnosed under the age of 50, if they're family members with bilateral breast cancer or also breast and ovarian cancer, more than 2 original tumours at different sites, if there are males with breast cancer, and if there's an overall autosomal dominant inheritance pattern. And so these would all lead us to think that there might be some sort of family history of transmission of BRCA1 or BRCA2 mutations. If it is discovered that a 30-year-old woman has the BRCA2 mutation, this has several implications, both in terms of treatment, but also ethical. So firstly, the patient might want to embark on screening, regular screening for MRI or mammograms annually from the ages of 30 to 50. MRIs tend to reveal a bit more detail. They tend to reveal a bit more detail and better at kind of defining the tissue. But either one of these can be used to indicate perhaps the development of a breast cancer forming. And another implication that might arise following the diagnosis of a BRCA2 mutation is the ethical aspects. So what we mean by this is if this 30-year-old woman who discovers she has the BRCA2 mutation, on the one hand, knowing this is kind of good, in a sense that she could perhaps inform patients and families, and then they can then have genetic counselling to understand how the results of this might impact them. And so this is important both at the individual and family level. And subsequently, this idea of testing of relatives is a topic which is highly debated. It has both its advantages and disadvantages. So for instance, through doing genetic testing, the advantages of these are that they can undertake appropriate preventative measures and lifestyle changes to perhaps minimize the risk of carcinogenesis and tumors developing. It can also inform the treatments. So for instance, if you have a known BRCA2 mutation, this might influence the way your breast cancer is treated. But the negative perhaps, or the common queries or the common concerns, with such genetic testing is it might actually increase the anxiety about getting the cancer if you know you're predisposed to it. And if you have a known positive result for, say, a BRCA2 mutation, there's a risk that there could be discrimination or problems, for instance, getting health insurance, although that shouldn't happen. It's something which quite feasibly could occur. And so, yeah, as you can see, the idea of genetic testing, both for the patient, but also subsequently for the patient's family, is a controversial topic, and it's good to be able to highlight the ethical sides of this topic. And finally, with our treatment options, if there's a known BRCA2 mutation, then this suggests the patient might be viable, or their condition might be conducive for PARP inhibitors. So PARP inhibitors essentially inhibit the PARP pathway, which is involved in single-strand DNA repair, which recognizes the exposed end after a single strand breaks. And so normally what happens is the BRCA mutations are involved in homologous recombination in double-strand breaks. And if a cancer cell has a BRCA mutation, it will then rely on the PARP pathway to survive. So if we can then knock out the PARP pathway too, this idea of kind of synthetic lethality occurs, whereby we've knocked down both of the critical DNA repair pathways. That means that the tumor cells are no longer viable. So an example of a PARP inhibitor is olaparib, and this has actually been recently approved for, or it has been approved for BRCA mutations, patients with BRCA mutations in, I think, ovarian cancer, I believe. And finally, if a patient is known to have a BRCA1 or BRCA2 mutation, they may want to have risk-reducing surgery, so a mastectomy or an oophorectomy, so removal of the breasts or the ovaries to reduce the risk of cancers occurring at these sites. And so you can see that this question itself is quite open-ended, but it's very broad in the sense that you need to consider the ethical implications, the therapeutic implications, and ultimately the factors that might be lead you to actually suspect the transmission of a mutation of BRCA1 or BRCA2 in the first place. So I think with that, we are done with the lecture today. I'll go back to the original slide, sorry, where you can find my email address. So here's my email address and name. So if you want to contact me, then please do. Yeah, and once again, thank you all for coming. If you have any questions, then feel free to just email me at any point. And if you are able to fill in the form, that would be amazing. I'd love to kind of hear your feedback and see what I can improve better for next time. I'll hand over back to Harry now.
That was brilliant. Thank you very much.